In this video, we will continue our discussion about UGGT1 and UGGT2 specific substrate. What it means that what are the specific substrates for which UGGT1 and UGGT2 acts or work, and we have discussed the general overview and initial part of the experiment in our last video, and we will continue from that side. In the last video we have understand that how they have planned their experiment to identify or the substrates that interacts with UGGT1 and UGGT2 both. However, they move one step forward, and now they have identified or they have designed experiment in a way that they will be able to identify what are the substrates that interact with UGGT1 and what are the substrates that interact with UGGT2. So in order to get an information about UGGT1 interaction, what they have done is, instead of just eliminating ALG6, which prevent the glycosylation in the dolichol phosphide, which is usually triglycosylated. So the first glycosylation in that N-glycan initial N-glycan that attached to the polypeptide chain is consisted of triglycosylated structure. Glycnac the complete structure is N-acetylglucosamine 2, mannose 5, and 3 glucosylated glycan. However, with the knockout of ALG6, now the glycans can no longer be glycosylated. So whenever in our analysis we get monoglucosylated glycan, that is because of, in this experiment, because of UGGT1, as UGGT2 is also been knocked out. And in the second experiment, what they have done is, to get the information about UGGT2 substrate, they have done similar to the first thing, but just one change, that is they have ALG6 knockout, and along with that they also did UGGT1 knockout. So they will get now the information about UGGT2. Before that, it's been considered that UGGT2 does not have glycosyl transferase. However, the result of this experiment shows that UGGT2 also act as a glycosyl transferate, and there are 3 specific proteins for which UGGT2 acts. However, other 30 proteins that are identified are overlapping with UGGT1. Let us understand and the result in details in the next slide. These are the results that have been presented in the paper, and first one is about the interaction of UGGT1 and its substrate, as it is ALG6 and UGGT2 knockout cells. And we can see that different in the y-axis we can see that different proteins that are been affected by or that interacts with UGGT1, and this color indicates their final location of the proteins. And for UGGT2, we from the first impression, we can see that the amount of substrate that interact with UGGT1 is more than UGGT2. Yeah, there is a spelling mistake, it should be UGGT2. And however, we can see that most of the proteins are yellow in color. More precisely, 45% of the proteins are yellow in color. That is, they are targeted for lysosome. So 45% of the proteins that interact with UGGT2 are having their final distillation in lysosome. And this C figure is about the this description of the UGGT1 substrate that and where are their final location. So we can see that in total it has identified 66 glycoprotein that interact with UGGT1, and uh, the major portion of that glycoprotein is of the plasma membrane, as we can see that, and next one is the lysosome. After that ER, then the secreted protein, and it is a multiple protein, and one is the nuclear membrane protein. And for UGGT2 substrate, we can see that maximum portion is been taken by lysosome protein. It's around 45%. Total number of glycan it is identified is 33. And next position is of the endoplasmic reticulum that has 6 glycan, and there is a tie between endoplasmic final proteins location is at endoplasmic reticulum and at plasma membrane. It is at both of them stands at 6. Next position is of the secreted protein, and then 2 multiple proteins. This D figure is about the overlapping of the glycoproteins that are identified in UGGT1 and UGGT2 knockouts, and we can see that there are just 3 proteins or glycoproteins that are unique or that are specific to UGGT2, whereas there are 36 in the UGGT1 specific proteins, and 30 of them are overlapping. And third one is about the quantification of the proteins that are present in both the experiment. How they have did the quantification is they have divided the glycoproteins that are as from the previous video we have understood that during the experiment they have TMT tagging, so they have divided the proteins that are abundance of the proteins that are present in UGGT2 specific substrate divided by UGGT1 specific substrates. So we can see that there are 3 specific glycoproteins that are abundance in UGGT2 specific substrate, and there is one protein that is highly abundant in UGGT1 specific UGGT1 specific substrate, that is CD164. And there is one more information available in this graph is that those which are represented in a circle are the soluble protein, whereas those that are represented in square are transmembrane protein. So soluble all these 3 proteins that are highly abundant in UGGT2 specific substrates are soluble protein, whereas the proteins that are highly abundant in UGGT2 specific substrate experiment are more is transmembrane protein. So that's all for this video, and I hope you have some insight about the result that has been presented in the paper. For more detailed information you can refer the paper that, and the link of that paper or the reference has been provided in the description. Thank you for your time.